All right, here we are in Chapter 5, Part 3, talking about large biologic molecules, sometimes called biomolecules. And we have a section on nucleic acids, of which DNA and RNA are the predominant members. So we're going to talk about basic structure, talk about some of that function, some of the functions you already know, but hopefully we'll introduce you to some stuff that maybe you hadn't heard about before. So virtually everybody knows about genes, and I can actually remember the time when the first time I heard the word gene used in a genetic context. I'm like, you inherit genes like blue genes? I think I was probably in second or third grade. But the gene is a functional unit. It is actually more than just the information. The gene actually contains elements that control when and how it's expressed, and there may be other sequences in and around the gene that regulate when and how it's expressed. And that's a later chapter in the book. But nevertheless, it's good to know that the gene is a sort of a functional unit. That's a sort of functionable, heritable unit, if you will, that confers traits. So genes are made of DNA, a nucleic acid, and the monomers are known as nucleotides. This is actually a nucleotide monophosphate. Okay, nucleotides usually have 3 phosphates. But in any case, you're going to want to be able to recognize these. There's a nitrogenous base, a sugar. In this case with DNA, it's deoxyribose. That's to say there's an OH here. And if there's an OH here, it's ribose, but you don't have an OH here. You have, it's just a hydrogen. So the deoxyribose has only one OH, and then we have 1, 2, or 3 phosphates. Outside of some rather exotic molecules, really the 2 major types of nucleic acids that we see in biologic systems are deoxyribonucleic acid, otherwise known as DNA, has an H here instead of its counterpart, the ribonucleic acids, which have OHs at both of these carbons. And by the way, the numeric convention stands if this is the, shall we say, the 0, the oxygen, 1, 2, 3, 4, 5. These are 5 carbon sugars. Oftentimes they'll put a prime next to it. So for instance, this is 0 again, 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Should get to know that system sooner or later because you're going to be using it at some point. DNA does provide directions for its own replication. The structure of the DNA and the way that it is present in the manifestation that it's present in the cells allows for a replication mechanism. It can also direct transcription, that is to say the conversion into another nucleic acid. So we start with one nucleic acid and we're turning it into the language of another nucleic acid. That's transcription. And when we turn nucleic acid into a peptide sequence, that's translation. And then that leads to protein synthesis. That area right there, these 3 statements or 3.5 statements, is collectively known as the central dogma of life. We have not discovered any life anywhere that does not follow these rules, that does not have nucleic acids at the heart. That is to say, DNA transcribed to RNA translated into protein. As far as we know, life obeys this central belief, this central dogma, because we just don't know. We're trying to figure that out. Along those lines, if you've ever seen the movie E.T., there's a moment, and for somebody who's involved in molecular biology and so forth, it is by far the most profound statement in the movie. Yes, there are more emotionally charged moments, but if you understand the central dogma, there's a moment when they're analyzing the extraterrestrial. He's called the Old Botanist, if you ever read the book, where they say, he's got DNA. That is so insanely profound that another life form from another planet, perhaps another galaxy, has DNA would be just absolutely mind-blowing. So Just indulge me a little bit there. I know, but that's what makes science fun sometimes is when you see references in popular culture. Here's the central dogma in sort of visual graphic form. DNA, deoxyribonucleic acid, is usually in a double-stranded, we call that the double helix, if you will, state, but it will open up transiently to allow for transcription. mRNA, which is the messenger RNA, and there are actually 3 different forms of RNAs, is created when we do that transcription process, and that mRNA is then exported, sometimes very short distances. I mean, literally just barely through that pore, or sometimes maybe a little further out into the cytoplasm. But either way, that mRNA leaves the nucleus and is then interacted with a special kind of protein structure that we're going to break out. When we get to chapters later on in the semester, near the last week or two, we're going to be talking about this. That ribosome then translates. So transcription is when we keep it in the nucleic acid family, shall we say, and translation is now we're turning it into the language of proteins. We're going to use the ribosomes amino acids to make these polypeptides, and along with the folding, the chaperonin stuff that we talked about last time, we're going to be making functional, hopefully functional proteins. Great video, by the way, worth your time. Use your resources. If these lectures are a little too dry for you, just hop on YouTube and give it a shot. If you have questions, just let me know. I'll be glad to say, hey, that's great, or hey, I don't know about that. So the components of nucleic acids. Yes, they are called polynucleotides, but nobody ever uses that word, but here we are. Okay, that is to say that they are polymers of nucleotide monomers. Nucleotides include a nitrogenous base, just depicted as base here as a square, this 5-carbon sugar, and I'm sorry this doesn't have an oxygen here, but there should be an oxygen right where my laser pointer is right now. You can see the oxygen here, and you go clockwise, 1, 2, 3, 4, 5. And you put a prime by it, 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime, and then 1 or more phosphates. So a nucleotide has all 3 of those functional groups covalently bonded together. This right here is the key with the nitrogenous base is that you're going to see this alternating carbon and nitrogen, or nearly completely alternating carbon and nitrogen. That's where the word nitrogenous base comes from. This one just happens to be a purine. We have purines and pyrimidines. The portion of the nucleotide, if you are missing the phosphate, if you just have the nitrogenous base, whichever one that is, of the 4 or 5, you have the sugar, but you don't have any phosphate, they call that a nucleoside. Some things here in this figure. Here's a nucleotide. Okay, it has all 3 components. Again, 0 here. 1, 2, 3, 4, 5. That orientation is very important because 5 prime, the 5 carbon, is considered the upstream end of the polymer. Just like the amine group in a protein is considered the upstream end. Most of the time when we're synthesizing these nucleic acids, we're synthesizing by adding nucleotides to the 3 prime end. Said another way, we're synthesizing 5 prime to 3 prime. And so what we're going to be doing is adding nucleotides to this OH. Again, dehydration synthesis. We're going to get rid of a water when we do this. And we're going to be synthesizing in this downward direction relative to this figure. Again, we got the purines and the pyrimidines here. That is the heritable information, if you will. That sequence of bases is going to tell us how to make an organism and how that organism is going to function. So here are the pyrimidines. These are just, again, nitrogenous bases, single ring. Purines have a double ring. Adenine, guanine, thymine, and cytosine. Those are the ones you find in DNA. What you find in RNA is that you switch the T for the U. But here are the sugars, and here are the bases. I'm not going to ask you to recognize which base is which, other than maybe I might say purine or I might say pyrimidine, just for conversational purposes. You got to look it up, you can. But it's a good idea. Purine rings are a double ring, pyrimidine rings are a single nitrogenous base. Just a few factoids about the structure of DNA and RNA molecules. RNA usually have only one phosphate backbone. They very, very rarely will form a double helix or a double phosphate backbone molecule. It does happen, but it's unusual. mRNA, ribosomal RNA, tRNA usually have only one phosphate backbone, one strand, if you will. Doesn't mean they won't bind, the bases won't bind together and form a hairpin or something, but it's one phosphate backbone. DNA, on the other hand, has two phosphate backbones, and they're wrapped around each other, called a double helix, but they're also what we call anti-parallel. That is to say, if we're going 5' to 3' down one strand, let's say this is the 5' end of this strand, and we're heading to 3' down at the end here. The other strand is going from 5' to 3'. This time 5' is here, going down the strand like that to 3'. So you'd have a 5' end on one strand and a 3' end on the other close together. That's what we mean by anti-parallel. And one DNA molecule usually includes many, many genes. And let me show you the picture in the next slide. This is a donut of double-stranded DNA shown in an abstract form here, somewhere around 16,569 nucleotides. This is the strand of DNA that we find inside the mitochondria of our cells. And usually, most of the time, the material in this mitochondrial DNA you inherit from your mother. We used to think it was exclusively from your mother, but now we find that mitochondria will share from time to time, and you'll get a little piece of your dad's stuff in here. Very rarely, almost 99, the vast majority is going to be maternally inherited. So you're a little bit more your mother than your father, if you know what I mean. In any case, this is a loop of DNA. It's a circular DNA molecule. And what we see here are the individual genes. I think there's around 24 or so genes, and there's even one on the other strand here. You can see most of them are on just one strand here. But there's one on the other side. And we have mapped this out, and we now have identified certain mutations in this mitochondrial DNA. Remember, we're more alike than we are different. So many of us have nearly identical mitochondrial DNA. It's very, very similar. In any case, though, there are discrete mutations at different sites that are associated with different kinds of cancers. So if you're interested in cancer, this is a great direction to go in understanding molecular biology, because that's where the action is and how cancer is formed and how it's treated. With those anti-parallel DNA strands, you have what we call a given pairing that goes on. That is to say, A always pairs with T on the opposite strand, and G always pairs with C on the opposite strand. And by the way, it's always a purine and a pyrimidine, We call that complementary base pairing. Okay, so here's our T binding with A, and it binds in 2 hydrogen bonds. C binds with G, and that has 3 hydrogen bonds. And this will make sense later on when we say that when you have lots of G's and C's, it takes a higher temperature to melt and separate those strands. What I mean is melt I mean separate them so they're no longer hydrogen bonded. So here's another role for hydrogen bonding. We saw it in water. We see it as important in the secondary structure of proteins, and now we're seeing it really important in the forming of what we call complementary base pairs in DNA. It can also occur between 2 RNA molecules or between parts of the same RNA molecule. Big difference in RNA is that you don't have T. You don't have the thymine. What you have is a close cousin of the thymine called uracil. And so A's and U's pair, again, with two hydrogen bonds. And here's a molecule of single-stranded RNA that's binding complementary to itself. It's making what we call a hairpin. But two different RNA molecules can bind to. Those usually don't last long. Those usually get destroyed pretty quick in the cell. And here's just the figure from your book showing you the anti-parallel nature, the sugar phosphate backbones, the complementary base pairing with hydrogen bonds. And here's a single strand. If you follow that blue line all the way around, I would have loved to have been the artist who did that. It's all one strand. It's just that it's folded back on itself, and nucleotides in that strand are forming hydrogen bonds to make this unique and specific structure of what we call a transfer RNA that we'll get to later on in the semester. And in addition to the idea that these things are functional in cells, they can also serve our ends in understanding science and understanding what's going on in our universe, that we can actually use these as a quote-unquote tape measure of evolution. DNA is incredibly durable. In fact, we're harvesting DNA from things like amber, and it can withstand even relatively harsh treatments like dry cleaning and so forth. That DNA is usually very, very well preserved across generations. But there are some changes from parent to child, usually in the tune of 1 in a million, which means that you have about 2,000 or 3,000 just small changes, usually silent changes in your DNA from your parents. We can see a combination. This is a combination of data here, but when we see very similar species that we believe are related, oftentimes you'll see more overlap in what we call sequence identity. That is to say, identical sequences in those two species. Let me give you an example of what I mean. We have sequence identity. Human beings have sequence identity with bananas to the tune of something like 16%. So out of 100% of our DNA, 16% has identical sequences, give or take, identical sequences to bananas. If we were to look at our sequences for us compared to, say, chimpanzees, which we believe, chimpanzees are a hominid ape, what we would see is a 98% identity with chimpanzees and humans. And you can see that if you pair up the genetic sequence, oftentimes with the structural similarities between two species, you often see that the DNA correlates with a structural relationship as well. So there's multiple lines of evidence that indicate that DNA drift over time is often associated with how related or unrelated two species are. That's what we mean by evolutionary kinship, if you will. And here's a figure to sort of back this up. This comes from the National Human Genome Research Institute. Like I said, bananas, maybe it's up to 18%, but or 16%, maybe in weeds it's 18%, 26% with yeast, 44, almost half of our DNA is identical to fruit flies, 92% compared to mouse, and that's why mice can make good facsimiles if we're studying biology. That's why mouse models are very popular often. Chimpanzees, 98%, so that molecular kinship, if you will. With that, we've completed Chapter 5. As always, if you have questions, just give me a holler. We'll do our best to help you out as best we can with the material if it's confusing or so forth. And thank you for your time again, and I will hopefully talk to you again soon.